Welcome everybody to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension, Wisconsin 4-H. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you Alyson Fleming. She's with the Department of Forest and Wildlife Ecology. She's here to talk with us about whale tails, ecological histories recorded in whale tissues, past and present. As I pointed out in my little missive this week, we are 600 and some miles as the bird flies to James Bay, which is the closest salt water.
That was a new fact for me. I did not know that.
Oh, yeah, that's all I'm good for. If you drive to Washington, D.C., which is the closest salt water, it's still about 840 miles. And it amazes me that we get to have scholars that are working on whales. So now I'm going to talk, ask you the 5 questions that I ask everybody. Alyson, where were you born?
Boston.
Massachusetts?
Oh, right. There's a Boston here too, right?
I have no idea.
Oh, there's something, I feel like there's every city in Wisconsin. And country.
And then where'd you go to high school?
Belmont High School. It's the public high school in town.
And then where'd you go to undergrad?
Tufts University.
And where's that?
Medford, Massachusetts.
Which is a suburb of?
Boston.
Thank you. We have a Medford in Wisconsin too, because my dad taught there. It's up north of Wausau. Yeah. And then where'd you go for your advanced degrees?
To Scripps Institution of Oceanography, which is in San Diego, California.
Okay. And what was your PhD in?
Yeah, in biological oceanography from there.
So Scripps is that part of UC San Diego?
Exactly.
And so you've got the Birch Aquarium and Museum there,
right. Yeah.
Which is my most envious thing ever. It's a splendid place to have as part of the university. And then when did you come here?
Just under a year ago.
Very
we moved here last July.
Good. Well, I'm looking forward to your talk. As I said, I think this is the first one we've ever had on whales in the 16 plus years of Wednesday Night at the Lab. So would you please join me in welcoming Alyson Fleming to Wednesday Night at the Lab.
Thank you. I'm excited to be part of the record breaking on whales here in Madison. So today I'm going to talk to you about a few different stories that whales tell. And that's why I've called my talk Whale Tales, because they really do have a lot of information to tell us. And as you can see, some of the pictures I've included are from historical artifacts. So I use a lot for my research. I use a lot of specimens and samples from museum collections around the world. And one of the last times I gave a talk about my research here at UW-Madison, students afterwards actually came up and told me about the museum that's right here on campus, and that there are actually whale samples here, which I did not expect and hadn't even looked into yet. So I haven't followed up on that in detail, but I plan to soon. And so please share any Madison, Wisconsin related whale details you might have afterwards, because I'm still new to that scene here. So I don't want to break your little cursor button. I see it's on its last legs here. So I want to start today talking about humpback whales, and they've been the subject of a few different research projects of mine. Humpback whales are the best studied of all the large whales, and that's because they're very cosmopolitan. They're often found near the coast. When you go on whale watches, they're a very common sort of sight that you'll see on whale watches. They're very gregarious. They often come close to ships, and they have a lot of above the water displays, more so than other whales. So a lot of the large baleen whales will just be traveling at high speed and take a quick breath for a moment, and all you'll see of them at sea is just their back for a brief moment and their blow. But humpback whales often are very active at the surface, and so they've been the subject of a lot more research than all the other large whales. And this image is a sort of cartoon map of all of their different migration paths in the North Pacific. And this was the outcome, one of the outcomes of a very large multidisciplinary project that happened over 5 years, and it was called SPLASH, which stands for Structure of populations, levels of abundance, and status of humpback whales. And it was international in scope. It involved researchers from Japan, all over Russia, Asia, and then of course, all down the US West Coast and Mexico and Hawaii in order to put this map together. And the way that we make most of these connections and have for about 30 or 40 years is just by taking photographs of the underside of the whale's tail. And so whenever you hear this term whale tails, it always has two meanings because it's the stories that whales tell, but also the underside of their flukes are really important for identifying individuals. Each of them have a pattern on the underside of the flukes that's just like individual, like our fingerprints are. And we can use those photos to then match. If we see one in Hawaii, look through thousands of photographs with many interns, and then find the match for that same individual in Alaska, maybe that year, maybe 10 years later. And over time, you can put together a picture like this, which shows you how these different populations all around the Pacific are connected and at what level. And so from this image, I wanted to start here because I think it shows you a little bit of background of how wide ranging these species are, how much of the ocean they truly move through. And if you think about sort of ways that we can be monitoring the ocean, these are in fact actually sort of samplers of the ocean because they're moving through so many different habitats. And they also do this every year usually. So something about their timing of when they do this, how many of them do it, when are they having babies, what are they eating, all these different things we can look at, and it can tell us something about the whales themselves, but also the oceans. And that's really been my area of research focus, is asking what can we learn from marine mammals and especially cetaceans or whales specifically. And in addition to whales being wide ranging over such large areas, they also are, they also are, of course, top predators. And sometimes they're feeding at relatively actually low trophic levels on krill, and other times they're feeding higher up on things like sardine and anchovy and squid, even lobsters in some places. And so they're integrating across these really wide areas of whole ocean basins. They're also integrating from top to bottom of ocean food webs. And they're also participating in moving nutrients and basic compounds around the ocean. And that piece is really sort of a newer part of research that we started to learn really how important at an almost biogeochemical level whales actually are. And for a long time, we thought that whales were really just sort of, not very numerous and probably, really cool to look at, but not actually very ecologically important. And over time, there's been a few different whale stories that have emerged from the research that have shown us that actually whales, though low in number today, are in fact ecologically very relevant and important at scales from biogeochemical levels all the way up to sort of levels of populations. And so I wanted to start by just telling you two quick sort of whale bedtime stories. And these are stories that you learn as a graduate student and hear over and over, whether you're studying whales or krill or any part of sort of oceanography, you hear these. And so one of them takes place in Antarctica. And this image is kind of a schematic summary of the overall story, which is a story about, my image is sort of in the way a little bit for you guys, but a change that occurred in the Southern Ocean ecosystem as a result of whaling, as a result of the removal of so many large whales. The thought is that the removal of all those top predators who were exclusive krill feeders released krill from competition. It removed their predators, so krill really expanded. And then that krill was available for other predators like smaller whales and penguins. And this hypothesis is really never able to be fully tested, but it's been discussed and proposed and studied in the literature for 40, 50 years, and it's called the krill surplus hypothesis. And it's still debated today, but it was kind of this revelation of, I think actually, when we remove the large whales, they do matter in the ecosystem. When we change them, other parts of the ecosystem follow and also change. So that was one of these sort of stories that pointed to the idea of the importance of whales in the ecosystem. And another one is another sort of famous story in oceanography, which takes place off the coast of California in the kelp forests of California. And some of you maybe have gone, has anyone gone and seen the sea otters off of California? one or two. And so the sea otters are a really charismatic species. They were also hunted very extensively. And as they started to have a rebound in their population after hunting had ceased, things were going well for the sea otter. And then all of a sudden something switched. And there were less sea otters. And as a result of less sea otters, there was way more of their prey, which was urchins. And then urchins typically graze down kelp. And so the kelp really disappeared when these urchins sort of became overabundant due to the removal of the sea otters. And it was after quite a few years of research, it was determined that this may have been the result of this whole sort of cascade of effects. It's called a trophic cascade, may have in fact been initiated because of changes in killer whale predation in the area. And they estimated, the authors who wrote this paper estimated that maybe just a handful of individual killer whales changing their patterns and their behaviors and sort of their dietary preferences and getting a taste for sea otters may have actually changed that whole system. So those just a few very large predators that consume large amounts, they would have been almost like, otters would have been like popcorn in a way, you know, like not very calorific, but very numerous, and this would have changed quite quickly. So both of these stories then showed us really whales matter, and they might play an outsized role in the ecosystem compared to their actual levels of abundance when we measure them from scientific surveys. And so what has been really rewarding and exciting for me in my own research is starting to delve into all of the technologies. This is well suited as I was looking for this building. I saw it said genetics on the outside. All the different technologies that have started to be developed in the last 10, 20, 30 years that we can now apply to whale questions in new ways. And so those include genetics, of course, also hormones. We can now study hormones inside of whales from their skin tissue, from their baleen, from their blow even. We're now using drones to capture moisture from the blow and measure hormones inside the blow. So you're not even harming the whale at all. We're using isotopes, which is the subject of most of my research. So those are stable isotopes, which I'll explain in a little bit. And those tell us things about what animals are eating and where they're going. And also pollutants. So there's now just this sort of suite, and this is not the exhaustive list, but there's so many different things that we can look at now when we look at whales for them to tell us both about what's happening internally with them as organisms at the level of their populations or at the level of the ocean basin, using them as sort of samplers of the oceanographic environment. And each of these different methods, I want to just take a moment to explain in a little bit more detail how we use some of them, because they can, depending on what we're using them for and what we're using them on, they can tell us different things. So these refer to different time scales that we can look at when asking questions about whales. So on the x-axis here, I have time or half-life of hormones, and then on the y-axis, isotopes. And they're very similar. But essentially what we're looking at is that there's all different types of matrices. What type of tissue, in other words, are we using from the whale tells us a different view into their life. So just like us, our skin tissue turns over very quickly, a matter of days, weeks. And so if we take a piece of skin tissue from a whale, that is telling us about a short amount of time in their life. If we go to the other end of the spectrum, like teeth or earplugs, which is like the sort of waxy core from inside of a baleen whale's ear, those are lifetime records. And so those can then tell us decades of information from one whale, and usually at a more annual time series, so more like tree rings. And so depending on what your question is, you can match your question and your technology to then the type of tissue, and you can ask questions about genetics and hormones and isotopes across different scales and across different individuals. You can combine methods. So it's really opening up so many different questions that we can ask about whales. And so to start off today, I want to tell a story about California. So back to the home of the sea otters. And this is again a humpback whale story. And these are humpback whales, as I said, are very well studied. They're also one of the most numerous abundant whale off of the West Coast. And National Oceanic Atmospheric Administration is mandated, as they are protected species, all whales are protected species, they have to be basically counted and kept track of all the time to make sure that their populations are doing okay and stable or growing. Excuse me. And so every 3 to 5 years, there's a huge, usually 4-month-long survey in every part of US waters. So NOAA has to cover all of the US EEZ, which means out to 200 miles offshore across all of our different coasts, East Coast, West Coast, Gulf Coast, Alaska, Hawaii. So it's a lot of cruises. That's what a lot of your tax dollars go to for NOAA research, is a lot of time at sea by biologists counting whales. And so these are 3 images of 3 different years, 2001, 5, and 8, of humpback whale distributions in the California current ecosystem. The black dots refer to actual sightings, and then the more rainbow colored are showing you densities of predicted information based on habitat preferences. So where do we think are sort of their most critical habitats? You can see, of course, if you look across those years, it differs every year. So unlike terrestrial environments, everything about the ocean continuously moves. So if you go and say, oh, this is a spot where whales always will be this year, next year, it will look completely different. The oceanography there is different, the upwelling, the winds, it all changes all the time. It's extremely dynamic, and it's in 3D, it's dynamic. So when we look at these kind of images, we're trying to be able to predict eventually, and now we're starting to be able to do that, where will whales be when the conditions are X or Y? That would be very helpful for minimizing interactions with ships, with fisheries, with entanglements, and understanding what are we going to see in terms of changes with global change in the future. So at this, with this kind of image, one of the questions we're really asking are, what are the physical and biological oceanographic processes that the whales are responding to that make them move in these different ways in different years? Can we understand those sort of underlying drivers and therefore really understand what is moving these whales? And so I want to do a quick little 101 on isotopes. Probably if you guys are, I'm sure this has been maybe covered in these kind of lectures before, but stable isotopes in ecology, as I mentioned, are really useful for understanding things about what the whales are eating and where the whales are going. And so probably all of you remember back from chemistry in high school, carbon-12 is sort of the most typical form of carbon. You'll remember that from the periodic table. And it's 12 because there's 6 protons, 6 neutrons. And in actuality, this is just one species of carbon. There are multiple. And so carbon-13 is another somewhat commonly occurring species that is also stable. And it is the heavier isotope because it has 6 protons still, but then it has just 1 extra neutron, 7 neutrons. And that makes it the heavier version. It's a little bit higher in atomic weight, but 99% of the carbon out there is carbon-12 and 1% is carbon-13. And this becomes helpful in ecology because when in metabolic processes, any metabolic process, whether it's inside of an animal's body or happening in sediment in the bottom of the sea or in phytoplankton at the water surface, all metabolic processes tend to be lazy and they'll preferentially use the lighter isotope. And so if you think about this inside of our bodies, for example, whatever we eat, we will preferentially use the light isotope. Using it means digesting it and also excreting it, and the heavier isotope stays behind in our body. So we accumulate over time, the same way you think about bioaccumulation for something like mercury or other things, we will accumulate the heavier isotope over time. And so we can then sort of use this as a tracer of ecological processes in the environment. And so you frequently will see for isotopes a plot like this, which is often referred to as isospace, where you have carbon on the bottom and nitrogen on the y-axis. And as you move up the food chain, you'll see this progression from the bottom left of the graph to the top right of the graph, meaning from lower trophic levels have lower nitrogen. And as you move up, you're getting more and more of that heavier nitrogen. So you're moving further up the graph. Carbon, so nitrogen really tells you a lot about trophic level. With each step in the trophic food web, you move up about 2 to 3 parts per mil in nitrogen. There's phrases for you are what you eat plus 2 or 3 in terms of nitrogen isotopes. For carbon, it's you are what you eat, usually plus about 1. And carbon tells us more about where you are rather than what you're eating. So trophic level is for nitrogen, and then carbon is more like, are you inshore or offshore? And over time, we've started to be able to sort of put different relative relationships on this graph. So are you in a more productive or less productive environment? Carbon can tell you something about that. Offshore, inshore also varies with carbon. So we can use these as tracers as animals move around. And so this is a map of about 300 humpback whale biopsy samples. And when I say a biopsy, what we're doing is taking a small piece of skin tissue from the whale in the wild using a modified crossbow. So instead of a pointy tip at the end of the crossbow, it's a hollow tip, and it's about the size of my pinky, or like the end of a pencil here. And it goes about that deep into the whale, and then bounces off. It has a little stopper and bounces off. And then we collect that bit, we can freeze it and use it for about 5 or 6 or more studies over time. And it can last in the freezer for decades. And so I then used 20 years worth of skin tissue that had been collected by NOAA Fisheries from 1993 through 2012 as part of a dissertation project of mine. And so the samples were collected from all along the California current ecosystem. And once I ran those for isotopes, what we saw was, this is again showing you that isospace plot with carbon on the bottom and nitrogen on the y-axis. Hopefully these numbers aren't too small for you to read, but the different colors are grouping the samples by year. So as I started to look at the data, it was clear that different years had different signatures. And there were sort of these different buckets of time. And so there were early years that were in the light green, then a middle set of years, 2004 to 6, that were in the more salmon color, and then 2010 to 12 are in the teal. And so you start to see these different groupings, and the most likely explanation of why you see changes in isotopes, especially with a whale like a humpback whale, who are very flexible and foragers, is that they were switching what they were eating. But with isotopes, because they also refer to changes at the base of the food web, or where the whale was eating, what the whale was eating, it's always sort of a few possibilities. And so what was great about the California current for this study is that humpback whales are the best studied large whale, and California current is probably the best studied of all the large oceanic ecosystems. We have a ton of data there. There's been multi-decades long full-scale surveys that are done 4 times a year and collect data on all sorts of prey and fish. And then we also have a ton of physical oceanographic data on temperature and climate indices. So I could look at lots of different levels of the food chain to see what was going on. Why were those isotope signatures changing year to year? And so I combined data from humpback whales, from sardine levels of abundance, from anchovy levels of abundance, from krill levels of abundance, and then a variety of other physical and climate indices. So things like upwelling and winds and temperature, El Nino, and a few other climate indices that are similar to El Nino, which are called the North Pacific Gyre Oscillation and the Pacific Decadal Oscillation. Those are other big climate regimes that switch. That's what that bottom graph is. And we know that all these things have a lot of variability. They have a lot of variability seasonally, annually, interannually. So it gets quite messy when you're trying to kind of take a picture away from this. And what ended up being really useful with the whales is that they were synthesizing across these large areas, as we talked about with that first map. And they also were synthesizing across numerous trophic levels and within a season. So by collecting a small piece of skin, we were looking at a few weeks of time in one foraging season for these whales. And what emerged is this story from the humpback whale's perspective of what was changing in the ecosystem. So in the first part of the time series, there was a lot of winds, high upwelling at the right time. There's spring seeding that happens just like we have on land. There's also sort of critical spring times in the California current that have to do with the amount of wind being there at the right time to support the primary productivity that happens in the system. And that was really good in the early years and in the late years. So we had helpful, productive upwelling, a positive North Pacific Gyre index, which refers to also the sea surface height and temperature. And in those years, there was high krill populations. And so we saw that the whales were feeding low on the food web, feeding mostly on krill. And that then in the middle years, everything really switched and the whales showed a higher trophic level. There was more sardine and anchovy in the system, not just more, but those fish populations moved in their distribution as to where they were to make them more available to humpback whales so that they were kind of lined up both in space and time. And this was also a very warm year. It was more stable in the California current. There wasn't as much wind. And so from this study, it was really helpful that we were able to use the whales, but then actually drill down and look from top predators to their prey, to physical and climate oceanographic indices and see kind of how all of that lined up. And this is very helpful because often we have sort of all of these disjunct. We're monitoring so many things at species level or at one particular index, like a temperature index or a wind index. And so we have a lot of data that will look like that bottom graph, but then trying to put it together as a story and understand what's really driving it is often harder to do because we don't have always the right amount of data or coverage spatially or temporally. And so in this case, we're able to sort of tell that full story and understand that full trophic system and what those whales were responding to. And I don't think I included this image, but what's nice is that I'll just scoot back for 1 second. If we look back at these maps, these years actually correspond to the early period, the middle period, and the late period with regards to those changes in isotopes. And so you can see that in the middle period, 2005, there's really quite a different distribution of where the whales were. And so that ended up being their fish year. And they were distributed further south. And in the krill years, there's often krill hotspots in the California current that will be just leeward of very strong upwelling systems. And those are focused around Point Conception and off of the Bay Area. And you see then why these distributions are occurring and they match up with what we've now learned about their prey. So the isotopes were very helpful in sort of resolving a lot of these connections. And so to me, this was an interesting study, a helpful study in sort of confirming the fact that whales really can be good marine ecosystem samplers and also integrators. They really can be these storytellers of what's happening in the marine system. And we now have such a diverse and expanding set of analytical tools that we can apply to this, that the possibilities of what questions we can ask using this kind of approach are really quite endless. And so this was all using modern samples from the 90s and 2000s. And it made me wonder if I could actually use that same kind of approach using historical archives, because of course, we as humans have had a very long period of interaction with whales. We've been whaling them since the middle of the 1800s, actually probably much longer. We're learning now more and more about active whaling being done probably even in medieval times. And so there are many, many records. We used marine mammal parts also for so many different things, right? Baleen used to be used for corsets and umbrellas and all these things that we use plastic for today. Often baleen was used. And we have images like this from, a long period of time. So we have drawings. We also have, of course, collected whales. We have hunted whales and kept some of their parts for long times in museums. So from all of these interactions, I was particularly interested in whether we could use this type of approach, especially to study in polar environments, which are inherently very difficult to study. We often have trouble accessing them. This is one image, I think, that kind of shows that without so many words. And we know that they're also the site of very rapid change on the planet. So they're sort of, we need to study them, and yet they're really hard to study. So can we apply the same kind of approach, but using museum collections or archive samples? And so these are pictures from the Smithsonian in DC, which I didn't know happens to be the closest body of saltwater from here. Good to know. But there are warehouses outside of DC in Suitland, Maryland, which, if any of you remember the end scene of Indiana Jones, where the ark is being put into the wooden box, and then you're zooming out and zooming out and zooming out, you just see boxes upon boxes upon boxes. That's like what this place is. And it's just massive amounts of boxes and cabinets, and 2 whole warehouses plus 1 whole other cube is totally dedicated to just whale parts. And these warehouses are like the size of football fields, and there's thousands of pieces of whale in there, teeth, baleen, skulls, whole specimens, jaw bones that are, reach the ceiling, just really massive collections. So there's kind of endless material to use. And so in those collections, there are also these pictures actually here with the writing on them. Those are Bowhead baleen plates, and you can see maybe some of the writing on there is saying New Bedford. That's actually where my family is from, and New Bedford was the largest whaling port in America, in the world, actually, for one period of time. And so those were caught by a ship out of New Bedford and then eventually ended up back at the museum and still have the sort of literal records on them to prove it. And so using those particular plates, as well as specimens from 12 other bowhead whales, I was interested in asking whether we could use bowhead whales as an indicator of Arctic change. What was happening in the Arctic when they were caught? And do we have enough samples now today to sort of build a time series and look at change over time? And bowhead whales are a useful study species for this question because they're the only endemic baleen whale to the Arctic. They reside in the Arctic all year round. They have this interesting figure 8 migratory pattern where they move in the winter, they're down in the Bering Sea in the pink. And then in the springtime, they're migrating, that sort of fluorescent green line is showing their spring migration north into the Beaufort Sea, where they hang out there in the dark green areas for the summer, and then follow the mustard color in the fall back down to the Bering. And so they're really good samplers of this whole area, which is all sort of this polar environment. And additionally, they have the longest baleen, or maybe second longest, of all whale species. And baleen is really cool as another type of that tissue that we're sampling, that matrix, like we were talking about with skin. But baleen grows over time, just like our hair and nails, and it's made of the same material, keratin. And it doesn't last the entire lifetime. It's not like a tooth or the earplug, like I mentioned, because it does wear down at the tip, but it includes many years of growth. And so this picture, maybe this is the pointer, yeah. So this picture is a bowhead whale that was caught by Inuits up in Alaska, and it's upside down on the beach, just to orient you. So this is the top of the head, and the baleen only grows from the top jaw and faces downward. And so you're seeing this animal inverted. And what I think is nice about this image is you can see, first of all, these individual baleen plates, they're called, and they're very hard. They're really kind of feel like plastic. They're bendable, but very sturdy. And they have a bristly end. It's just sort of the fraying side, and that acts as a broom, as a filter for catching their prey. And then the water gets pushed out through the spaces in between. Good image of that in Finding Nemo, if you remember that scene. And so the other cool thing about this photo is, can you see these lines kind of horizontal going along here? I don't know for sure, but I'm pretty sure these are actually annual growth layers of the baleen. And so that tells you a little bit about how many years are in this baleen, just as kind of a visual for that. And so then we could drill these baleen plates at 1 centimeter resolution to look for what is the biochemistry in the water that these animals are sampling over time. So it's acting like a tape recorder through the life of the animal. And so this is one of my interns from a few years ago, Catherine Louie, in the Smithsonian collections, drilling this very long 3-meter piece of baleen. So I think we get here, maybe it says over 200 samples from that, because it's 3 meters. So we're getting even closer to 300 samples from these, just with a Dremel tool that you can buy on Amazon or Home Depot. And we make little scores in that every centimeter and collect that powder, and then run that powder for carbon and nitrogen isotopes. And from that, we get images like this, which is really sort of the full length of the baleen summarized then for isotopes. And the blue is nitrogen, the green is carbon. So this is now a different orientation to those other graphs that I showed you with the carbon on the X and nitrogen on the Y. Now we have length of the baleen, which is basically time, and then nitrogen and carbon. And you can see that there's these very sort of regular cycles and quite short. And so each of these cycles is 1 year in the life of the animal. And so their changes in isotopes change because they are moving in that figure 8 pattern through different biochemistry backgrounds between the Bering and the Beaufort Sea. And as they're eating, they're incorporating these different signatures into their baleen. And so we can use this to learn something about them again and their habitat. And so, yeah, just to orient you, this is the point of capture here, closest to the gum line. And then the oldest material is farthest from the jaw at about 300 centimeters out. And if we take 12 of those, so this is one individual, and their more negative values are the Beaufort Sea, and their more enriched values are the Bering Sea. And then if we take all the individuals in the collection and know their years of capture, and then do the isotopes on them and see how many years we caught of data in each of those baleen plates, we can then start to put together a time series of all of the different plates together. And so the earliest ones we had in the collection were from the 1850s. And then Inuits are still hunting a handful of bowhead whales every year. So we have also some samples from the 80s, up to the late 80s. And so putting this all together for looking at carbon, we can see that over time, over about 150-year time series, we had this decrease in carbon. And so it was pretty level up until the 1950s or so. And then we have a gap in samples. And when we pick them back up again in the 1980s, we see that the carbon is quite low. And this information here about Seuss corrected. So because of us as humans burning burning fuels, we're actually putting lighter CO2 back into the atmosphere. So we're changing the carbon pool in the environment to be lighter isotopically than it used to be. And so we always want to correct for that based on what we're comparing when we're looking at these long time series. And that's called a Seuss correction. And so even if we account for that lightening of the isotopic pool in the environment, which is this dotted line, you can see that there's still quite a decline from the 1950s to the more modern samples. And there's a few different reasons why this might be occurring. And the same pattern of a decline in carbon has been seen in other samples. So it wasn't too unexpected, but part of that might be because one reason is because the sea ice has been retreating earlier and lasting longer. There's sort of longer summer seasons. And so the whales might be spending more time up here in the Beaufort Sea each year. And this is isotopically more negative or lighter isotopic environment up here compared to their wintering habitats. And so more time in that area could be possibly lightening their isotopic signatures. Another option is that the carbon signature of open water phytoplankton, open water algae versus ice associated algae, which forms the base of food webs in all marine systems. The carbon signal for the pelagic algae, the more open water algae, is lighter, and the ice associated algae is more enriched. And so over time, if we're losing ice and moving towards more pelagic or more open water, less ice dominated environments, you would also expect to see a decline in the carbon. So those signatures were kind of made sense. They were explainable, but the nitrogen signal was this. It was just kind of all over the place, not something dramatically different at the start or end of the time series, a bit messy. But we decided to use an additional tool to look at this, which is called compound specific isotope analysis. And it's looking at individual amino acids. So far when I've been talking about carbon and nitrogen isotopes, you're looking at all the different amino acids in the sample. Now there are tools to look at individual amino acids like leucine, and there's maybe 12 or so typical ones that we look at. And so when we're talking about carbon and nitrogen so far, we're referring to what's called the bulk. So that's looking at the nitrogen and carbon ratios of light to heavy in all of the amino acids. But when we do compound specific, we're choosing one amino acid at a time and able to look at each of those individually. And what's cool is if we separate the different amino acids, some amino acids are what we call source amino acids, meaning we get them from the base. They're made only at the very base of the food web. So like only in the ocean, only by phytoplankton, or on land, only by primary producers like plants. And those are maintained no matter how many steps they go through in the food web. Whereas other amino acids are called trophic amino acids. And those change, just like we already talked about with nitrogen changing as it moves up the food web. And if we use both of these from one sample in a whale, you're essentially getting a view into historical times about both what the whale was eating and what the base of the food web looked like. So it's as if we have phytoplankton samples from 1850, which obviously we don't normally. And so this is very helpful. And from those isotopes, what we found was that something very surprising, which is that in the early part of the time series here, close to the 1870s or so, 1860s, the phenylalanine, which is one amino acid, which is the source amino acid, meaning that's telling us about the base of the food web, that was quite low isotopically. And I thought going into this study that the big change we would see would be recent years, just like the carbon, right? This would be a story about climate change. Something is changing in the Arctic system in the last few decades, but that didn't turn out to be the case. The big change actually was here. And we can compare that to also other amino acids, which tell us about the trophic level. And the trophic level changed a little bit here, but stayed more stable than the base of the food web did. And it was mysterious to me why the base of the food web would change very dramatically in the late 1800s and not change very much recently. We know that huge ecosystem changes are happening. We've been fishing massively. We've changed structure of numerous ecosystems in the marine environment. That's been really well documented across multiple trophic levels. So why would this be stable in the last 50 years, but dramatically different in the first few decades? And so thinking about what this phenylalanine is telling us, when we see low nitrogen phenylalanine signatures, what that actually is indicating is that there's usually high nutrient availability for nitrogen. And so if we look at what we know about nitrogen today, the surface water content of nitrogen or nitrate around the world, this is obviously a large scale map of the entire world's oceans. The red colors, the warmer colors mean more nitrate at the surface. The cooler colors mean less. And if you think about where the bowheads are, they're here, and they're moving annually from up here in the summer to down here in the winter and back in. So they move from quite high nitrate environments to lower nitrate environments. And so if you keep that in mind, and then we are thinking, okay, what else was happening in the 1800s? I looked into information about mercury levels and information about output from the, there's a large Mackenzie River there, which changes how much flow and fresh water. We know that can change things about nitrogen and carbon. We looked at changes in climate, all these different things. And what we came to conclude is that actually one of the biggest changes that was happening was whaling itself. And so this is a map of all of the captures of bowhead whales. And what was kind of miraculous about the bowhead whale fishery is that it started and ended very quickly. It only lasted for about 40 years, maybe more like 60, 49 to 14. And there was a real temporal change. So you can see that these are color coded. So the green dots are where the whales were caught in the early years of the fishery. And as the fishery continued and the decades progressed, the whalers had to go further and further into the ice in order to find whales. And that was because, of course, there were less and less whales, so they had to travel further. They wouldn't have done that if the whales were still able to be caught here. That was, of course, very risky to go into the sea ice environments where they're shipped. It was a lot more time, which meant money. And so they had to keep going further and further north. And then very quickly, sort of just as the fishery started, it also ended. And so you can compare kind of these early years, not just spatially, but also with catches. And so keeping in mind where this sort of big change in the phenylalanine happened, we can look at those particular years with regard to the capture data for the whales. And so this is all of the data from 1864. So the sort of earlier part of the maybe 20 years in, 15 years into the fishery. And you can see where the whales are being caught, and that would be consistent with this time point. And then a few decades later, where the whales were being caught was a totally different spot. And there were also almost no whales left at this point. And there was basically the entire population of bowhead whales was removed from that ecosystem within 45, 50 years. And so that really dramatic change, when I first looked at this data, I thought this is, again, back to the sort of beginning of my story, do whales really matter? We hadn't really had that much information to say thus far that at a biogeochemical level, whales would be making a difference. Like, why would we see different nitrate levels in the system across that different 50-year time series? And now, like 10 years later, we have actually many more papers that say this actually really is probably consistent with what we've learned by doing other types of sampling with whale systems, and that whales actually are a really important part of what's called the carbon pump. And sometimes you'll see in the news now about whales being important for something called blue carbon, which is storing carbon in the oceans in various ways. And so we now know that whales move nutrients throughout the system. So they feed in one place, like they feed in the Bering Sea, they migrate, they're taking with them their nutrients, they're feeding often at the bottom and stirring up nutrients, and then not doing that at other places. They're migrating then north and sort of being like a nutrient highway in a way when they're existing in these different parts of the ecosystem. They're also vertically integrating different amounts of nutrients. So they're feeding in one level of the ocean and then swimming up to a different part and pooping in that part, which releases tons of nutrients in one area. And so they really are the same way that hippos and other large predators we now know are quite important for redistributing nutrients across terrestrial ecosystems. We're finding out that whales do the same thing. And so the removal of that many whales that quickly from quite a actually small ecosystem, this is a very shallow area of the ocean, that many whales may have actually been the reason why we saw those really big changes in nitrogen and biogeochemistry levels from the early 1850s to the 1900s. And now when we plot some of that data with the return of bowhead whales coming back in pretty good numbers, we actually start seeing the nitrogen return to what it was at the beginning again too, which is sort of further support for maybe there is something to the role that these animals are playing in moving nitrogen and other nutrients around the system. And then one quick support for that is just to study other side of the country in the Gulf of Maine, measuring actual nitrogen flux in the Gulf of Maine. And so they measured it today, looking at different nitrogen inputs to nearby coastal waters in the Gulf of Maine. So they compared rivers, coastal point sources, atmospheric deposition of nitrogen, and whales. And so you can see that whales here are not nothing, right? This is today's numbers when they're relatively reduced. And compared to atmospheric deposition, they're quite low, but they're more than rivers and pretty comparable to coastal point source areas of nitrogen deposition. And then they did models to look at what they might have been historically. And so there were low, medium, and high versions of the model. Like if we had this many whales, what would the numbers have been like? If we had only half that number of whales, what would the number have looked like? And so from their, even just their low scenario of what we think historical populations of whale numbers were, that low scenario puts them at more than atmospheric deposition today. And so thinking about this in further detail and using modeling approaches, we might start to understand a bit more, especially in these kind of contained ecosystems like Gulf of Maine, which is a very sort of circuitous circulation pattern that retains a lot of nutrients, or up in the Gulf of Alaska, similar. We might see that the role of whales in some of these ecosystems is really important in maintaining that productivity. So that's where I'll stop. And yeah, thank you to all the museum samples and everyone who's helped there, and to many different places where I've run samples and processed specimens over the years. And thank you for having me.
Questions?
Great talk. I'm
Thank
interested
you.
in your first slide.
Uh-huh.
Maybe you didn't study this and other people did, but so do the same animals go at the title slide, migration slide? That way.
Yeah.
Do the same animals go to the same locations, and do they interbreed at the summer feeding, and then where would the offspring go?
Yeah, great question. So they do go, we know that they go mostly to the same places every year, and they learn that destination from their mother. So the calf follows the mom for the whole first year of life. So calves are born in the more tropical areas here. These are the breeding areas. The calf will follow mom for the whole first year of life, learn the feeding area and learn the way back before it separates from them. And there are sometimes change-ups, but I would say like 90% or so are matching back to their same location every year.
So you're getting species populations.
Exactly.
That are separate.
Exactly. And that's actually what this study, the SPLASH study, was exactly asking, was how many populations are there? So looking like drilling down at how many movements happen between these breeding areas. So these black connections are moving from breeding areas, which then obviously would be a mixing of populations. Whereas if each breeding area only sourced one feeding area and then had consistency going back to that breeding area every year, 100%, you would have a totally exclusive population, which might reach species level. But at this point, we see 5, I think, different populations in the North Pacific.
I have one more quick one.
Yeah.
Who gets the ear sample?
Yeah, great question. There's a scientist, Steve Trumbull, who I think he's at Baylor University, and he's done a lot of the most work on the earplugs, and he's mostly looked at hormones, and those studies are really interesting too, because some of those studies have looked at whether or not whaling and wartime has stressed whales out. So looking at the stress hormones over these, 70, 80, 90-year time series in some of these whales, he's asked those questions of like, do you see big spikes in certain years, and what else was happening in the ocean at that time? Yeah.
What is the lifespan of whales?
Yeah.
What's the lifespan of whales generally?
Yeah, it varies. So for all the whales I talked about today, they're all baleen whales, and those are the ones that we typically think live the longest. So humpback whales are probably around 60 years or so. Bowhead whales, the second part of our story today, are the far end of that spectrum and probably can live, of course, we don't know how many of them do, but can live over 200 years. Yeah, and we know that because of a few different, there's a cool technique for looking at the eyeball that tells you how old the animal is, but also from archaeological evidence of finding arrowhead embedded deep in the tissue of one of the whales that was then caught by the Inuits in like the early 2000s. And that arrowhead was dated back like 150 years. And the individual would have already been, more than a calf when it was hunted. So yeah, pretty incredible.
Other questions? The yellow population is owned by Japan. Japan notoriously has been
Whaling.
Scientifically whaling. Do you use their data? How different what's the kind of data that they collect in their rather large scale whaling scientific?
Yeah, that's a great question. So historically, they had North Pacific whaling, and then the more recent contemporary whaling that we continue to hear about is all focused in the Southern Ocean. So actually, I have a different project that's looking at Japanese whaling, baleen from Japanese whaling expeditions after World War II. But the North Pacific, there's been a lot of efforts to work with Russian scientists, actually both Japanese and Russian, but there was a huge period where Russian whaling was very poorly documented purposefully. And so there's been real efforts to work with both Japanese scientists and Russian scientists to verify and validate all of the records and sort of rework them so that they're correct. And that's been helpful for understanding things like how many whales used to be in the ocean. If the whaling data is accurate, we can better back project how many there used to be. So
forensic angle to some of this?
Exactly. And then also some of the contemporary data does give us information about things like stomach contents and stuff that usually we're not collecting, obviously, because you have to kill the whale to do that. So we do try to at least use some of that data that comes out of those programs. And do you want me to, I saw there was some in the chat.
Yeah,
Should I open that? Okay. Did the washing away of man-made debris all the way from the coastline of Japan to the California coastline after the last earthquake affect the health of the whale population, and does any research data support it? That is a good question from Raj. I do not know specifically about the debris. We definitely do see, with connection to this earthquake, we do, a lot of the whales that end up on beaches today do have a lot of plastic and trash and pollutants in their bodies. That's for sure, from everything from small cetaceans and pinnipeds, seals, sea lions, to large whales. So that's definitely a problem, but the extent of it is something that we don't have a ton of scientific data about, because it's all really sort of dependent on just the animals that wash up on the beach. And that's kind of in of itself a bit of a biased data sampling method. So there has to be quite a lot of data points in order to then start understanding at population level scales how much of an issue is plastic for these populations being able to subsist for decades more.
Well, which earthquake is he referring to?
I'm guessing maybe, yeah,
Okay.
yeah. But yeah, I don't know specifically that we've seen anything really with connection to whale populations from that.
Other questions? So how accurate was the study?
It was actually quite accurate. Yeah, it was a fan favorite of many marine science grad students that I know, especially the songs that teach you the zones, you know, which zone you're in the sea. Very handy when you had to memorize that anyways for class. But yeah, pretty accurate, and more so than Pinocchio. So that was helpful to kind of rewrite that.
So starting last year, the sea shanty came out.
Uh-huh.
Can you walk us through what they're talking about in that song?
No, I don't know if I know the one you're referring. I mean, there's like a whole world of TikTok that's
Billy
all
O.T.
sea shanties.
The Billy O.T.
Oh no, I don't know this.
Something sugar and rum. They talk about tonnage, only it's not weight, it's okay.
Sorry, blank stare. No, yeah, that probably would help, I'm sure, yeah.
I'll pull it up later.
Yeah, we can analyze it in detail.
Any other questions? If not, thank you very much.
Yeah, absolutely, thank you.